All right, today we are diving into a massive shift happening in oncology. We're looking at how two fields that seem totally unrelated, the gut microbiome and artificial intelligence, are coming together to completely rewrite the rules for treating breast cancer. This is all about moving away from one-size-fits-all medicine and into an era of true precision therapy. And it's happening right now. So let's start by getting a handle on the sheer scale of the challenge we're up against.
First, just to put this in perspective, an incredible 80% of all breast cancer cases are diagnosed in women over the age of 50. So let's be clear, this is not some niche issue. It's a huge public health challenge that absolutely demands better, more effective solutions.
And that brings us right to the heart of the problem for anyone in this field. Chemotherapy is still a cornerstone of cancer treatment, but its effectiveness, it's wildly unpredictable. The reality is, every single patient is different. You've got this incredible patient heterogeneity, different tumor types, and of course, drug resistance. All of this means that for any given patient, the outcome is often a dangerous gamble. And solving this unpredictability is exactly what the industry is racing to do. So, where on earth do we find a solution? Well, believe it or not, the answer is coming from a place that very few people expected: the gut. There is a mountain of evidence growing every day that points to our gut microbiota as this hidden but incredibly powerful player in deciding whether chemotherapy is going to succeed or fail.
So what are we even talking about when we say gut microbiota? Well, it's this vast ecosystem of trillions of bacteria, fungi, and viruses that live in our digestive tract. For literally centuries, we just overlooked it. But now we're finally understanding that it's basically an indispensable organ that is co-evolved with us and is deeply tied into our body's most critical functions, especially our immune system and, crucially, how we process drugs.
And the data here is just so compelling. When researchers actually compared the gut bacteria of healthy people to those with breast cancer, the differences were just stark. As you can see, healthy guts tend to have way more bacteria like Coprococcus and Anorostipes. But in patients with breast cancer, you see a much higher abundance of things like Bacteroides and Feingoldea. This isn't just some random correlation. It's a clear biological signal telling us something important is going on here. But here's the thing. This relationship is not a one-way street. Not at all. It's this dynamic, two-way interaction. Chemotherapy affects the gut. And then, in turn, the gut affects the chemotherapy. And you know what? Understanding this feedback loop is the absolute key to unlocking its massive therapeutic potential. So first, let's look at what chemo does to the gut. These powerful drugs, they're not surgical scalpels. They're more like sledgehammers. They cause a lot of collateral damage. They can wipe out microbial diversity, they fundamentally shift the whole community of bacteria that's left, and they totally disrupt the production of critical metabolites. Those are the small molecules our bodies need to function. It's a massive disruption. Okay, now for the really crucial part, what the gut does in return. And this is where things get truly revolutionary. The specific makeup of a patient's microbiome can directly influence how a chemotherapy drug is metabolized. It can literally determine if the drug will work at all. It can dial the body's immune response to the treatment way up or way down, and it can even play a role in how severe those devastating side effects are. All of this has given rise to an entirely new scientific field: pharmacomicrobiomics. This, right here, is the new frontier for personalized medicine. It's all about understanding and then leveraging this gut-drug interaction. And for the life sciences industry, let me tell you, this isn't just some interesting academic curiosity. It's a huge strategic area for future growth. But think about it. How in the world do you make sense of trillions of microbes and billions upon billions of data points? The complexity is just mind-boggling. There's really only one way to decode all of this, and that's with a tool powerful enough to see the patterns that no human eye ever could. And that tool, of course, is artificial intelligence. And look, this isn't just a theory. As the research paper puts it so clearly, AI enables the identification of microbiota-derived biomarkers. So what does that actually mean? It means AI is the engine. It's the thing that can sift through all that noise to find the specific bacterial signatures that can predict disease, forecast how a patient will do, and guide a truly personalized treatment plan. So what does this actually look like in practice? Well, this slide is a perfect example. Researchers used a machine learning model, in this case a random forest algorithm, to try and predict how patients with metastatic breast cancer would respond to a common class of drugs. And the result? They got an area under curve, or AUC score, of 0.946. Now, just for context, a perfect score is 1.0. So getting a score this high, this level of predictive accuracy, is nothing short of exceptional. It's a real game changer.
Okay, so the science is fascinating, right? But what does it really mean for the future of treatment? I mean, knowing all this is one thing, but acting on it is something else entirely. So let's talk about the concrete, actionable implications for leaders in the life sciences space. The path forward is actually becoming pretty clear. First, companies have got to start integrating microbiome data collection into all of their oncology clinical trials, period. Second, they need to be developing companion diagnostics that can actually read a patient's microbiome to predict their response to a drug. Third, it's time to seriously explore therapeutics that actively change the gut, things like advanced probiotics or prebiotics to make chemo work better. And finally, use AI to mine all this rich data to find completely new drug targets. So the ultimate takeaway is this. We are right at the dawn of a fundamental shift in medicine. We're moving away from the blunt instrument of one-size-fits-all chemotherapy and toward a future of incredibly personalized interventions, each one precisely tailored to a patient's unique microbial fingerprint. And all of this leaves us with one final and I think very crucial question. The science is here, the technology is ready, and the potential is just undeniable. The only question left for every single leader in this space is this. Is your R&D strategy truly prepared for the microbiome revolution?